So hi, hello and welcome again. I'm Oliver, Microbehunter here. I just recently got an email with a question which I would like to share with you, and unfortunately I don't have a satisfactory answer for it. I'm just going to also demonstrate to you what the problems are. So that's a little bit the point of this video, to show you the difficulties of making certain slide preparations. Well, let's start off with a question first. A few weeks ago I collected some soil from a flower tub and placed it in a glass jar. I added water and a few drops of milk as a nutrient source and kept the jar undisturbed for about 5 to 7 days. When I later observed the water sample, I found some rotifers and ciliates in it. After watching a video, I became very interested and decided to prepare a permanent slide of these microorganisms. If you could kindly provide me with the step-by-step procedure for preparing permanent slides of rotifers and ciliates, it would help me a lot. Thank you very much for the question. Just a little bit background info here. By adding milk, a small amount of milk to a water sample, you're making a so-called an enrichment culture. You're basically multiplying, growing those microorganisms so that they're at a higher concentration. This is sometimes done. There's always the danger of overfeeding, of course, so the amount of milk or nutrients that you add has to be quite small. But I digress. Okay, so what I have over here is a water sample, and I got this water sample here actually from some moss. I've collected some moss over here, and I basically then sprayed some water over it and collected a little bit of the water here. And this sample contains quite a few ciliates and also rotifers. So in that sense, it's quite similar to the sample of my viewer who asked the question. And I've got two microscope slides here, and this one over here, you might actually see that I already put a small drop on here and it's now dry. And I would like to show you now the big problem of actually having a fresh sample and a dried sample, because if you want to prepare permanently mounted slide, you have to dry the sample. But let's do this first. I'm going to show you now how the original specimen sample looks like. You allow the things to settle to the ground here. You collect a little bit of material from the bottom. You put it on here. And what I'm going to do now is even without a cover glass, I'm going to show it to you how it looks like under low magnification. So the quality is not going to be good because there's no cover glass on here, but at least we're able to see what's present here at a fairly high concentration. Well, even if it's a little bit blurry, you can already see there's quite a bit of movement going on. It's blurry because the microorganisms are able to swim up and down inside the drop of water. After all, as I just said, no cover glass is on here, so I can only also look at it using a low magnification. But all of the living things are easily identifiable because they move around, right? Of course, there's also plenty of other debris present and plant material and so on. But there is movement going on, and this actually shows that the sample is well and alive. Now, if you want to make a permanently mounted slide, you have to dry it because many mounting media, of course, they have a solvent that evaporates, and this is already the first problem, because those water microorganisms are extremely delicate, and as soon as you dry them, they will start to shrink and they will deform. Some of them might even, if you add the wrong mounting medium, start to break apart into lyse, and then basically lysis means that the cells break open and the contents spills out, and that's a problem, of course. And in order to show you now how the dried specimen looks like after air drying for about 10 minutes or so, I'm going to show you that one. Again, here, this one is simply also without any mounting medium. Yeah, and this is basically the dry sample. And you, of course, no movement as expected. But if you just notice, it's very difficult, if not almost impossible, to actually see where there were cells. I mean, maybe we can zoom in here a little bit. Here, this is a slightly higher magnification, those 4 round oval-shaped structures. But I have to be honest with you, they do not look like cells, but actually like the so-called the shells or the so-called the tests of certain amoeba, maybe. Yeah, but as you see, these are probably not ciliates here. Yeah, but other structures. And the rotifers, I was not able to identify or find them at all because there are microanimals, and this basically means that they do not have a cell wall, and this also results in them losing their shape. And if you look carefully over here, you're going to see that there's a nice, interesting patterns, and these almost look a little bit like crystals that have formed because maybe any small amounts of salts or minerals that were present in the sample started to crystallize out. That's why you get those nice patterns. Yeah, here again at a slightly higher magnification. So you see, that's the problem. Drying actually kills them. Obviously, that's not the problem, but the bigger problem is that we're losing the structure of those organisms, and for this reason, it's very difficult to make permanently mounted slides. I'm going to show it to you in any case, just for the sake of completion, but do not expect any good results. Okay, so we're not going to see a great deal more. We'll be using also a difficult to use mounting medium, which is called glycerin jelly. I'll show you how that one works. So the mounting medium that I'll be using is glycerin jelly or glycerol gelatin, and this one is specifically made for pollen, because pollen are also quite sensitive to dehydration and to water loss. You notice that it is stained red. Well, a pigment has been added, a stain has been added. However, there are also versions without a stain. It doesn't matter for our purposes. It'll work fine. And this mounting medium is difficult to use because, as you can see, it is in a solid state. And what I have to do is I have to warm it up. And not only that, but I also have to warm it very carefully, because overheating it will actually also destroy it. I have to melt it, and then while it's still liquid, I have to add the cover glass and allow it to cool down. So this basically means I have now two options. Should I melt the whole jar or just a small sample? I highly recommend that you take a spatula and remove a small amount from the gelatin, and then place it directly on the microscope slide. So let's go ahead. Maybe I can already take this one here that's on the side here. That's going to be fine. That's already way more than we ever need. Okay, it's way more than we ever need. I just drop it on here. Okay, here we go. It's I think already too much a little bit, but for our sake it will be fine. So this here is my hot plate. It's set to 40 degrees, however, it's much hotter than that, so I'll turn it off and I'll place the slide with the dry water sample and the glycerin gelatin on here. And we're going to give it a few minutes and we're going to see when it melts. So here we go, and I've already got the cover glass ready. Okay, I might actually remove maybe some of the glycerin gelatin with my, you know what? I'll take my tweezers. I think this is already way more than enough. Maybe I can remove this. It's difficult to do that. Here we go. Cover glass goes on top. Let capillary action do the rest. We quickly remove it again, allow it to cool, and then now we're going to have another look and see if we're actually able to see more. Well, I will tell you already right now, no, this will not have regenerated the ciliates and the rotifers. So, well, this here, that is again the so-called the test or the shell of an amoeba. But looking through the whole specimen, we see that some structures slowly start to also stain pink because of the added stain, like for example this structure over here, right? But it too does not really look like a ciliate, because those that were present were actually much smaller. Rotifers I was not able to find. Of course, the occasional air bubble, because glycerin gelatin is quite a difficult to use medium because it creates so many air bubbles. And yeah, by the way, I'm using now my 40 times objective, so we should be able to see the cells without problems. But I think you'll notice already that due to the drying process, the cells kind of started to almost disappear a little bit, right? And therefore not quite as interesting as in the fresh specimen sample. So what do we learn? What do we learn from this? We learned from this that it probably is not worth it making permanently mounted slides from all specimens, especially if they're moving around, and especially if movement is such an important characteristic. Ciliates and other microorganisms are quite interesting to observe because of their movement. This is actually lost, and worse yet, due to the processing, we are not even able to find them. Now, of course, I did a little bit of research, and indeed there are possibilities of making permanently mounted slides of ciliates, but this is pretty advanced. You have to quickly kill them, you have to prevent them from actually shrinking and drying. You have to add some stain. So silver staining is commonly used, but this is quite advanced. But look at this here. We are a little bit lucky. This is a huge one, actually. Yeah, this is actually a rotifer that we're able to see here, but it's not quite extended anymore. I just show you this to you. Yeah, this one over here, is appears to be a rotifer, right? But again, unless you know what you're looking for, it's going to be kind of difficult a little bit. And in most cases, yeah, the interesting specimens, yeah, are not visible. Yeah. Glycerin gelatin is quite useful and very useful, and also commonly used for preparing algae. Also, a beautiful single-celled algae can be prepared this way. But yeah, the processing is actually a difficult, yeah, provides quite a few difficulties for for other specimens. Okay, again, the oval structure looks like the test of an amoeba. Yeah, yeah. I just wanted to share this with you, and simply encourage you to continue experimenting, of course, but especially if you're interested in documenting moving water microorganisms, I probably recommend that you start filming them and that you start actually documenting them using pictures and videos, because in a permanently mounted slide, it's a little bit of a problem. It's not so quite as easy. It's not quite as easy as I've just shown you. From my side, that's all I would like to share with you. Hope you found it informative. Happy microbe hunting as always, and hope to see you in the next video again.